Hello, everyone. Welcome to my video tutorials for spatial dataset analysis. In my previous video, I showed you how to analyze the Xenium human breast cancer dataset using the SquidPy package. In this video, I'm going to show you how to analyze the same dataset using the Seurat package. So you can download the dataset from the 10 times Genomics website. We can go to the 10 times Genomics website and have a look at the dataset. So you can see here is the link for the human breast cancer dataset. We are going to use the in situ sample 1, replicate 1 sample. So they performed the in situ hybridization for 313 genes, and they also performed the immunohistochemistry to stain the tissue with antibody CD20, ERB2, and also the DAPI staining to identify the cell nuclei. So we can scroll down to the bottom of the page. You can see here is the link to download the output files. After you download the files, it is a compressed zip file. You need to unzip the file. Then we can load the data into R. So you can see I downloaded the data and also unzipped the data. It is in my download folder. So this is the file name to save the downloaded data. The Seurat package has a function called load Xenium to load the spatial data from 10 times Genomics Xenium platform. So we can specify the files directory, and we named the field view as field view. C will be Xenium. If we run this code, we will generate a Seurat object, and we name it as Xenium object. Let's run the code to create the Seurat object for spatial data analysis. Okay, you can see we created a Seurat object. First, we can remove the cells that with 0 counts, because there is no point we can keep those cells for the analysis. So let's remove those cells. If we click the Seurat object, you can see at the moment we have 313 genes, 166,363 cells. So we can use the wiring plot to have a look at the n features and the n count, because we created a array as Xenium. So the features will be n features Xenium and the count will be n count Xenium. So we can run the wiring plot. So you can see we generated the wiring plot for the n features Xenium and count Xenium. You can see for the genes, most cells have detected genes around 60. Then for the total count, most of cells have the total count below 500. So we can subset the data according to the wiring plot. For example, here we can set the n features bigger than 5 and smaller than 200. For the n count, we keep the cells that at least have 10 count, and the total count below 1000. So the quality control we performed here will affect the cell clusters, because we want to see all the cells on the spatial coordinates. If you want to keep all the cells for the analysis, you can skip this step to subset the cells. For today's demonstration, we can subset the object according to the threshold we set here for quality control. As I showed you before, we have 166,363 cells and 313 genes. If we perform the subset now, and we can have a look at the Xenium object now, you can see we still have 313 genes, but now we only have 163,779 cells. So here are the final cell numbers after quality control for downstream analysis. So we load the data and pre-process the data. Now we can run Seurat workflow to cluster the cells. So we are going to use the improved normalization method, SC transform method, to normalize the data. If we perform the SC transform function, it normalizes the data and also scale the data. At the same time, it identifies highly variable genes. But for today's analysis, because in this data set we only have 313 genes, so we will not use the highly variable genes identified by the SC transform analysis. We will use all the genes because we know all the genes are in the row names of the object. So in next step, when we perform run PCA analysis, we set the features as row names, which means we use all the gene names as features and perform the PCA analysis. So after PCA, we can run UMAP. Now it is the standard Seurat workflow analysis. So after dimensional reduction analysis for both PCA and UMAP, now we can cluster the cells. First, we need to find the neighbors for the cells. Let's run. Next, we can find the clusters. We are going to use the resolution as 0.2 to identify the cell clusters. So now we finished the standard workflow analysis. We can use the dim plot to look at the cell clusters. We will use the group by function to see the Seurat clusters. So you can see we generated the UMAP for the cell clusters. Here we have 12 clusters in total. So in next step, I'm going to show you how to annotate the cell clusters. If you don't know well about the cell marker genes for your tissue, then you can run the find all markers function to find the mark genes for each cell cluster. Let's run the find all markers function to identify the mark genes for each cell cluster. You can see down here, Seurat is calculating the mark genes for each cell cluster. It started from 0 until cluster 11. So now you can see we generated a data frame named the wall markers. you can click the data frame and look at the marked genes for each cell clusters. You can see the row names are the gene names. We have the p-value, log2 fold changes. Here is the cluster name. You can use the marker genes here to identify the cell types for the cells in each cluster. So we can select some marker genes from each cell cluster and perform the feature plot. Because we only have 313 genes here, so all the genes will be among those genes. You can use the row names for the Xenium object to see all the gene names. You can see here are all the gene names. In total, we have 313 genes. So now we can use the feature plot and plot some marker genes for different cell types. For example, we can use MS4A1 CD79 to identify B cells, ITGAX to identify macrophages, and CD3E to identify T cells. Let's run those mark genes. You can see from the feature plot, MS4A1 and CD79A labels this cluster and this cluster. Then ITGAX label this cell cluster and CD3E label the T cell cluster. Next, we can run some T cell markers. We can run CD4 and CD8 T cell markers. NKG7 is the mark gene for NK cells, and KT is the mark gene for master cells. If we run feature plot for those mark genes, let's run it first. You can see from the feature plot, CD4 and CD8 enable different population in the T cell cluster. NK cells also enable some cells in the same cluster as T cells. Because we only have 313 genes here, and also we use the low resolution, so the NK cells and the T cells are in the same cell cluster. Then it labels the master cells in this small cell cluster. Next, we can run mark genes for like endothelial cells. It is PKM1. Myeloid cells, we use the KRT15. Then fibroblast, we use the NAM. We can also run the Ki67 as a mark gene to identify proliferating cells. Let's run it again. So you can see PKM1 label the cell cluster endothelial cells. KRT15 label the myeloid cells. Then NAM identify the fibroblast. Ki67 is largely expressed in this cell cluster. Later I will show you this is the cell cluster for the invasive tumor cells. Because this is a human breast cancer dataset, but next we are going to run some marker genes for the tumor cells. According to the dataset, the CEA-CAM6 is a mark gene for the ductal carcinoma cells. Then FASA is a mark gene for invasive tumor cells. Let's run both mark genes for the tumor cells. So you can see CEA-CAM6 label this cell cluster, and ASN label this cell cluster. So according to the mark genes and also the feature plot, we can name the cell clusters. Cluster 0 will be invasive tumor cells. Cluster 2 and 11 are the ductal tumor cells. The cells in cluster 6 are myeloid cells. Cluster 3 has fibroblast. 4 is macrophages. 5 is endothelial cells. Then in cluster 1 and cluster 10, we have both T cells and NK cells. 7 and 8 are B cells. Then cells in cluster 9 are master cells. So now we can name the cell clusters. Let's run. Because we named the cell types, now we can get the cell ident and create a new column in the metadata named as cell types. So let's create the cell types column and save the cell types information in the metadata. Because we named the cell types now we can use the group by function to count the cells by cell types. If we run the dim plot now, you can see we generated the UMAP plot. Let's zoom in. You can see now we named the cell clusters: invasive tumor, ductal tumor, myeloepithelial cells, endothelial cells, macrophages, fibroblasts, mast cells, B cells, and the TNK cells. So we named the cells. Now we can visualize the cells on spatial coordinators. First, we can set the group by function as Seurat clusters to see the cell clusters on the spatial coordinators. So here, the cells will be counted by their Seurat cluster numbers. So you can see we generated a spatial image plot to show the cell clusters on the spatial coordinators. Because we set the group by function as Seurat clusters, so here the cell clusters were counted by their cluster number. Next, we can use the group by function as cell types. Let's make the plot again. You can see now we counted the cells on the spatial coordinators as their cell types. So we can see the cell types on the tissue. So next we can visualize the expression level of genes that you are interested in your data analysis. Because this is a tumor data set, we can visualize the expression level for the invasive tumor gene FASN. So we can use the image feature plot function to visualize the gene expression level on the spatial coordinators. Let's run. We can zoom in. You can see FASA has a high expression in those cells we know they are tumor cells. So here you can see the FASA expression was indicated by the level from 0 to 3. The red color shows high level FASA expression in tumor cells. So next we can plot tumor-specific marker genes. I showed you on the feature plot for both ductal and invasive tumor genes. Now we can plot both genes on the spatial coordinators. Let's run. Okay, you can see now we have the cell types on the spatial coordinators. We can also see the invasive tumor cells and the ductal tumor cells because they are labeled by the blue and the red color. Okay, I'm going to stop for the demonstration. If you want to save the Xenium object, you can save it as the code I wrote here. Then next time, when you want to do more analysis, use this analyze the Xenium object. You can read it into R again and perform more analysis. So please subscribe my channel if you haven't to do so, and share my videos with your friends. Thank you.